Our next speaker is Jeremy Freeman from Genelia Farm. I guess it's Genelia Research Campus now.
Change the name.
So,
people were always confused why we were at a farm, because we don't do any farming. So they officially changed the name. All right. Put this thing on. Okay, great. I'm going to get started. My name is Jeremy. I'm a neuroscientist working at Genelia Research Campus. And I, like probably at least some people in the room, if not all people in the room, want to understand how the brain works. And at least for me, I do this by working very closely at the intersection between neuroscience, experimental neuroscience, and computation. I truly believe that there is a lot to learn about the nature of computation by studying the brain. But I also believe that at least right now, we basically have no idea how the brain works. Definitely not for humans, and even for a lot simpler animals like mice or zebrafish or flies. So I believe there's a lot of work to be done in just understanding how brain works. And I've chosen to do that by working at the interface between, on the one hand, experimentalists doing a variety of amazing experiments in neuroscience right now, and on the other hand, computation, which includes both the analysis of data, but also how to make sense of what the brain is doing, how to make sense of experiments, and how to design experiments. So everything I do is collaborative with experimental groups, both at Genelia and elsewhere, and that's what I'll be talking about today. I'm going to start by describing for you the kinds of experiments we can do, the kinds of data we can collect, which is pretty remarkable. There's a lot of amazing things that have happened in the last several years. I'm then going to talk about the kinds of things that we do with the data that we generate from these experiments, because that's become its own sort of exciting and interesting problem. After that, I'm going to give you at least one example of the kinds of things we're learning by doing these experiments and using these analytical methods, and then end with my sort of own vision for the future of where this kind of work is going and can go. So first, in terms of the things we can do, the kinds of measurements we can make, a lot of the work I do is with techniques in both, the ones I'll talk about are in zebrafish, and also mice, where we can use large-scale imaging of neural activity as mediated or indicated through a calcium indicator, GCAMP, which allows us to monitor the responses of large populations of neurons simultaneously in behaving animals. So one example of that, and this is in collaboration with a fantastic collaborator at Genelia, Misha Arens, whose lab has developed methods for monitoring neural responses from a larval zebrafish, this little guy here, using something called light sheet imaging, also developed with Philip Keller at Genelia. And this allows us to monitor calcium responses in the zebrafish brain, basically the entire brain, at single neuron resolution, while an animal simultaneously performs behavior. In this case, the behavior is for the animal to watch a grating or a little pattern underneath him that's moving by him, and we detect his intention to swim in response to this. Fish will swim to keep up with the movement of something underneath them, sort of trying to keep up with the world as it moves below, and we can pick up that and monitor his swimming behavior while monitoring neural activity and presenting the stimulus. So that all together looks something like this. In this movie, the brightness indicates a change in fluorescence, and this is a sort of indirect readout of neural activity at the single neuron resolution. The pattern here shows what the fish was looking at. This was the visual moving pattern. You can see every 10 seconds or so it starts to move, and the size of that circle indicates the animal swimming. So every time this thing starts moving, he starts swimming to keep up with it. So what you're seeing here is this confluence of complex dynamics of neural activity, a visual stimulus, as well as the corresponding behavior. And we can do all of this sort of regularly while exposing the fish to a variety of stimulus patterns as well as monitoring a variety of behaviors. So I also do a lot of work in another very cool model system, which is the mouse. And I'm highlighting here one of, I think, the most exciting mouse behaviors developed by a very close collaborator, Nicholas Sofrenio and Carl Svoboda, working also at Genelia. This behavior is a virtual tactile environment. So the animal, the mouse shown here, his head is fixed so that we can do imaging, and the animal is running through a virtual environment as determined by these two walls on either side of him. So he's running on a ball, and these two walls are moving back and forth, and the movement of these walls is locked to his running so as to simulate his experience of running through a corridor. So he basically thinks he's tracking the wall of an environment. And in the case of the mouse, we can't monitor responses across the entire brain. I should have mentioned the reason we can do that in the zebrafish is because it's transparent in its larval form. The mouse, of course, is not. as you can tell just by looking at it. But using 2-photon imaging, we can monitor responses over fairly large populations of neurons. In this case, we're looking at neural activity in the somatosensory cortex, the vibrissae somatosensory cortex, while the animal is performing this behavior, and start understanding how the dynamics and patterns of neural activity relate to behavior. All right, so those are the kinds of measurements we can make. And I should say there's a lot of very now new exciting technologies for not just monitoring neural activity, but manipulating it. And I'm going to be talking about that a little bit later. So that's the measurements we can make. But now we need to decide and know what to do with the data. So these kinds of experiments, these large volumetric imaging of neural activity, yields very large and also very complex data. I've heard recently, I think Nate Silver tried to distinguish between, I guess, big data and complicated data. These data happen to be both. They're big just because the raw size of a giant collection of high-resolution, high-temporal resolution images is pretty complex. But then there's also all of the richness to neural responses, sort of population activity, population dynamics, that we need to make sense of. The overall problem that we face and sort of the workflow that we try to achieve looks something like this. And this, I think, is not so different than a lot of other problems in sort of large data crunching fields. We have raw data, which is usually really big. From it, we usually want to try to extract signals like the time courses of individual neurons or other kinds of sort of summary or simplified representations of the data. And those are the things we then feed to a variety of different analyses to try to make sense of, for example, how neural activity is related to the animal's behavior, to what the animal was experiencing. That, in turn, we want to visualize, we want to share it, we want to explore it. And the really cool thing about neural data is that we usually have no idea how to do this. There's a lot of different methods we might try. There's lots of things we need to try in order to understand the structure of the data. So all these pieces of this process are really interrelated. Every time we do one thing, it sort of changes the way we think about wanting to do the other thing. For a long time, at least in neuroscience, the way this was done, sort of all of this was done, is basically single workstations in individual labs, with a lot of labs sort of independently coming up with strategies and solutions for doing these kinds of analytics. So we've been moving and really pushing towards a sort of newer, different way of doing things. And a lot of that emphasizes moving the analyses we're doing away from sort of single workstations and towards cloud-based tools and using distributed computing platforms. And in particular, building open source solutions that let us collectively as a community develop tools, share distributed computing, cloud-based approaches to working with these kinds of data in order to really share and kind of vet what we're all doing and make sure what we're doing is reproducible. So the particular things we're sort of looking for in building out that kind of platform are that we need our analyses to be fast. We need them to be interactive. As I said before, we never quite know exactly how to analyze these data. We're always trying lots of different things. And we want to do things in the cloud. I think the area of doing all of these kinds of analyses on the workstation in the lab is sort of over. So we have been particularly excited by and have been using now for a couple of years a very cool new technology called Spark. Some of you may be familiar with this. It's become exceedingly popular, largely in industry, as really a new platform that sort of supersedes and sort of complements, but also dramatically extends Hadoop for doing distributed computation on various kinds of data. About 2 years ago, we started exploring the use of Spark for neuroscience, and it's turned out to be a pretty phenomenal tool. It's grown a lot over the last several years and has been now used in all kinds of applications, but we use it to process neural data. Spark itself is really more a sort of library or set of primitives and a platform for working with distributed data and providing a set of abstractions to make it easy to work with distributed data. Spark itself is not built for neuroscience, so we have been building libraries on top of it that allow us to do the kinds of neural analytics that we need on these large imaging, largely imaging data sets that we're generating. So this library, which we call Thunder, as well as a visualization library, which we call Lightning, that I'll talk about at the end. These are open source platforms that use distributed computing technologies like Spark to do analytics on a variety of different kinds of data. Our general workflow is to start with data that can come from a variety of different sources. This is ultimately coming from the brain, and we try to bring it into a sort of ecosystem in which we have standard internal representations of distributed data on which we can then do a variety of manipulations as well as analyses, all very fast and either on clusters, compute clusters at universities or in the cloud, and then also come up with interactive ways of visualizing it, which I'll talk about at the end. This is really a project that started with some of us at Genelia, but has now grown, and there are at least 10 different labs starting to use this, as well as lots of people contributing new components and new functionality, which we're really excited about. So I think this is a kind of cool direction for how to take the kinds of analyses challenges that we face when working with this data. So I would say my lab spends a pretty big chunk of time doing this kind of stuff, but that's not really why we're not building it just to do it. We're building these tools because we want to understand how the brain works, and we want to use these things in the context of experimental neuroscience to both make sense of neural data and ultimately try to understand how the brain works. As I said before, I think we really don't know that much about how the brain works, and there's a lot to do. And what I want to walk you through now is an example of a combination of experiment and then a really cool manipulation that I think sort of highlights some of the kinds of things that we're able to learn, or we're starting to learn, and the ways we're thinking about probing neural systems. So this particular little vignette is part of a collaboration with Simon Perrone, a fantastic postdoc, and also working with Carl Sabota at Genelia. So Simon, long before I came into the picture, had been developing ways of doing very large-scale imaging in mouse reversal cortex. So Simon's looking, in this case, looking at a behavior. It's a lot simpler than that virtual reality behavior I showed before. Here we have a mouse who is using a single whisker, not even all of his whiskers, but just one. That simplifies things for reasons that'll become clear. And he's using that one whisker to detect the presence of a pole. So basically, is the pole at this position, or is it one of these positions? And if it's at the one position, he licks to the left. And if it's in the other position, he licks to the right. And you can train mice to do this. Simultaneously to the mice doing this behavior, Simon's imaging neural activity using GCamp systematically across a large fraction of somatosensory cortex, specifically almost all of the excitatory layer 2-3 neurons in a single barrel. So not over all of the barrel cortex, but just in a single barrel. This is the chunk of brain that corresponds to the representation of this one whisker that the mouse has, because the rest have been trimmed off. And with this method, we can record or monitor responses, not simultaneously, has to do this sequentially, but ultimately monitor responses during this behavior across about 10,000 neurons, excitatory layer 2-3 neurons, in the single barrel. So one of the things we then need to do with these kinds of data, along with a variety of processing challenges, is to relate the neural activity, which we measure, to properties of the input, or properties of the behavior, the stimulus. So this animal is using its whisker to detect the presence of this pole. There are a number of things about that experience that relate to or affect neural activity. And we break that down by fitting statistical models that relate basically the input to the output. So in this case, we describe the input in terms of two kinematic variables, which we call delta curvature and theta, and these capture what happens to the whisker basically as it moves and as it touches the pole. And we have these variables by monitoring using high-speed video tracking the position of the whisker, and then we can relate by fitting a model these different components of the animal's experience sensory experience to the neural activity. And for example, in this case, this lets us distinguish between neurons that are largely related to the delta curvature, which is something that happens as the animal's whiskers touches the pole, as opposed to just change in the position of the whisker. And we call these touch neurons and whisk neurons. So by doing this statistical calculation, we can basically categorize neurons as either touch in blue, whisk in green, or mixed in whatever that is, teal, as well as the degree to which they are reliably or faithfully encoding these variables across a fairly significant fraction of this piece of cortex. Again, this is the barrel, at least indicated here, that corresponds to the one whisker that the animal has. So this is, in some sense, a very systematic, very comprehensive description of how a piece of cortex is encoding some aspect of an animal's behavior. But one of the things you immediately, and I think this theme will show up a lot, you immediately start wanting to do is not just monitor all of this and characterize it, but now start perturbing it. So we need to know not only in what way are these neurons correlated to what the animal is doing, but what happens if we start manipulating it. And I think that is really going to be the secret to understanding the nature of the computation. So to take just an extremely simple, sort of deliberately dichotomized description of two ways that this piece of cortex, this population of neurons, might be encoding or transmitting information about the whisker touching the pole, you could imagine a sort of purely feed-forward system in which input from the thalamus to this piece of cortex drives the blue neurons, the touch neurons, And that's basically it. And that is largely the signal on which the animal is basing his decision. But you could also imagine, there are a variety of reasons I think this might be a useful computation, you might imagine forms of recurrent excitation among this population of touch neurons that would, I don't know, sort of recurrently excite and maybe strengthen the representation or enhance the representation of the thalamic input. So just by making the measurements I just described, we don't really know how to dissociate these. There are a couple things you might try, but one very direct way is to basically start killing neurons. So if you pick, for example, a blue cell and we ablate it, which is something we can do, then we can identify by measuring the effect of nearby neurons what the consequence of that ablation was, and it might tell us something about the nature of the computation. So Simon, also working with Freya, who was in the lab for the summer, developed a really sophisticated way of doing targeted ablations of individual neurons in the mouse. So in the overall paradigm for these experiments is that we do imaging of neural activity during the behavior I described. We analyze the data, so we have to do the analysis really quickly, basically over a period of a couple hours while the animal's still in the rig. Then Simon and Freya developed ways of ablating individual neurons using a 2-photon laser, and then after the ablation we image again to figure out what the neurons are doing after we killed a bunch of them. So this is a functionally specific manipulation, and just to show an example of how this works, over here I'm showing 3 neurons, and these are traces indicating their response to a touch event before they were ablated, and the first 2, 1 and 2, were selected for ablation, and the third was spared. And after we ablate, 1 and 2 have responses that are basically flat, so these guys are gone, whereas 3 is still responding. So we've taken these neurons, selected them based on a functional property, and then killed just a subset. So we can now ask, what does this do to the encoding across the population of information? And I'm going to start by showing you the control experiment because I think it makes the whole thing a little bit simpler. So in the control experiment, we deliberately pick neurons for ablation that weren't doing anything. So here is a map like the one I showed before of both the touch neurons and the whisk neurons, and the red are the ones we chose for ablation. And these were neurons that were basically silent, so they were doing nothing as far as we could tell. And after ablation, things basically look the same. So you can see that in the maps here. On the right, we're quantifying this by the count of neurons that had a particular level or score of either touch encoding or whisk encoding. And you can see these distributions before and after ablation are basically the same in the case where we ablated neurons that were doing nothing. And that was the nothing there was quantified based on a low level of activity or no relationship between the response and these variables of interest. If, however, we selectively ablate only neurons that showed a touch response, so basically only kill the blue neurons, then we see this very dramatic effect, which is that the consequence to the neurons related to whisking or involved in whisking, they're basically unaffected. These distributions look really similar, but the neurons that were involved in touch have been significantly suppressed. So by killing a subset of neurons, sort of removing them from the network, there's a selective effect on the rest of the network, but only for the neurons that were functionally sort of the same functional class to the neurons that we killed. So this is now a result that I think, first of all, gives some evidence, though it certainly doesn't prove, an idea that there's some kind of functionally specific recurrent excitation within this network that could provide a variety of computational benefits to the encoding of information. We don't know the answer to that yet, but that's obviously now what we want to explore. I think it also shows the importance of perturbation, that just by monitoring responses, there's a lot about what the computation is sort of what the role of the computation is, and the role of the dynamics and sort of properties of connectivity that are I think, best revealed or possibly only revealed through these kinds of perturbation or manipulation experiments. So I want to end by describing to you a little bit of sort of my own vision for the future, both where we're going in this kind of work, as well as I think some of the technologies and some of the questions that we're most excited to be answering and asking. And the first step forward really involves making everything I just described a lot more flexible and a lot more tactile. So when we're doing these experiments like the one I described, where we sort of image for a long period of time, then we do the analysis for a couple hours, and then we go back and do the manipulation, we want to have that loop be as tight as possible. And in the limit, we really just want to be doing the analysis of neural populations and of neural data while experiments are taking place. We don't want there to be any separation between sort of the experiment and the analysis. So what we've been developing, and this is with the help of a phenomenal guy working in the lab, Yasha Swisher, we've been building out a platform using the same kind of distributed computing technology, Spark, and specifically something called Spark Streaming, which is an engine for distributed stream processing. We are building libraries that let us take data, both behavioral data and also data directly from the brain, as it's being collected, and basically in real time or during the experiment, ingest it into a cluster which is doing distributed computation on the data and providing us with essentially online feedback as to what the brain is doing in a way that will scale to potentially very large data sets, as well as provide us with a lot of flexibility in terms of the kind of computations that we want to perform. And the idea here is that by doing those kinds of analytics as well as visualization online during experiments, we can do very tight closed-loop manipulations. Not only sort of perturb neurons on the basis of their functional response properties, but even adjust the behavior in which the animal is engaging or the stimulus based on the neural responses. So a big part of this turns out to be not just how to do the analytics, but how to do the visualization. Because if you want to watch a system, a complex system in real time, you need to have ways of looking at it. So that's a big part of what we've been developing. And in particular, we've been building out a platform, which I mentioned before, called Lightning, that's really a new way of doing visualization, sort of scientific visualization. It emphasizes a number of things, namely that visualization should be web-based. There's a lot of really powerful web-based platforms for doing visualization now. We want to take advantage of them. We want visualization to be flexible and dynamic. There are lots of different things you can do with data, and we want to make all of that available, as well as interactive. I think there's a huge opportunity, much of it really untapped, for how to use interactivity when doing scientific visualization, and we're trying to make all of that possible. So Lightning, I should say, a lot of it is the brainchild of Matt Conlon, an absolutely fantastic developer working with me. That's his Twitter handle if anybody wants to check in with him. And we've built this tool called Lightning, which is a server-based model for doing interactive visualization. The idea is that data can come from anywhere, but often it comes from an experiment or from some other source. Data comes into the Lightning server, and then the Lightning server renders that data using a variety of different web-based platforms and web-based technologies. So this includes things like D3 for doing interactive charts, but also libraries for 3D visualization and a variety of other things. With Lightning, we can render a variety of different visualizations that support a lot of the kinds of things we want to look for in these kinds of data, whether it's sort of graph information or time series information, sort of matrixy things, maps. That's just for fun. We obviously are not mapping the United States. We're mapping the brain, as well as a variety of ways of looking at images and volumes and doing lots of 3D rendering and all kinds of other things. It's really just getting started, but this is a platform that lets us build out a variety of interactive visualizations. And along the sort of in the same vein, we're working with a number of collaborators, Jean Gris, Tariqa Das, and again, Nick, where we're developing algorithms that work on streaming data and use both distributed analytics, but then also streaming versions of our visualizations to do various kinds of model fitting on data on the fly. So I'm showing here a little animation of a streaming clustering algorithm. I'm showing it in 2 dimensions, but of course the appeal here is that this thing scales to potentially very, very many data points in very many dimensions. And what's going on in these little pictures is that data are coming from a number of clusters, and these little black dots are tracking them. And these are the results of running this distributed Spark-based streaming k-means algorithm on the data as it arrives. And there's also a one-dimensional example to see a little bit more the way that these clusters are tracking the data. And so you at least one example of how this might be used for time series data, which is, of course, a case we're interested in. So this kind of thing we are now deploying in the context of rich, complex behaviors that we can perform in animals. This is one I started with, where the animal is performing this tactile virtual reality environment. It's exploring this corridor in real time, and it's sort of guided by the walls to explore this corridor. And while he is doing this, we can simultaneously be both watching the brain as well as analyzing the data as the data basically literally come off the microscope. So I'm showing what the data look like here on the left, and on the right I'm showing a map of a sort of model fit to neural activity. In this case, the color indicates the particular position of those walls that the neuron was most responsive to, so sort of tuning for wall distance, and the brightness indicates the degree to which the neuron was showing that kind of response at all. So what we get is a map where we can now say the degree to which individual neurons in this small population of somatosensory cortex are related to the position of the wall relative to the animal, or the position of the animal relative to the wall. And this allows us, or will allow us, to now make very targeted perturbations as well as changes to the behavior really online as the experiment is happening. We basically have this open in a web browser right next to the experiment, and we're just watching this as it evolves. So it's really I think a lot of opportunity to really change the way we do these kinds of experiments and guide these perturbations to move away just from these correlational measurements towards something more, more direct and more manipulative. And I just want to close by saying that, again, I'm very interested, very excited by the relationships between computation and neuroscience and what we can learn about one from the other. I think that's coming out of what we're doing in one way through the development of these new platforms and new technologies for data analysis that are really designed to work with neural data, but are actually sort of changing and sort of giving us new ideas about how to work with data in general. I also think it will go the other way, that we will learn things about the brain that will ultimately guide computation, but I think we also have a lot to learn about the brain. It's this incredibly complicated thing, and we're really just scratching the surface of what it does and how it does it. So that's really what we're trying to do. I want to highlight a group that we've started called CodeNuro. CodeNuro was a group really designed to start bringing together people from the neurosciences and people more from the sort of data science, tech, visualization world. We did our first event in San Francisco in the fall of 2014. It was really fun. It was very successful. A couple of people here, I think, were there. And we're doing another one in New York City in April 10th and 11th. And all the info for it you can find at CodeNuro.org. This group is about both throwing events. We also do hackathons where we work on collaborative projects to develop technologies and analytical tools for neuroscience. We also share data sets and make those available. So you can find out all about all of that at CodeNuro.org. And finally, I just want to give a couple links about the projects that I just told you about. These are open source projects for analytics and visualization, as well as emphasize really what we believe to be the future of this kind of work, which is that everything should be open source and the science itself should be open and reproducible. And with that, I will take a couple questions, I guess. Thank you, Zach.
Very fun stuff. Thank you very much. So when you're thinking about all the streaming stuff, it's really very natural for the immediate responses of all these neurons. But then when you think about the longer time scale of the adaptability and the task dependence and all of this stuff, I mean, the data is going to be there on the back end because you've streamed it in the past. But how do you think about bringing it all into the future and thinking about how the past affects the present?
Yeah, that's a great question. So there's sort of, I think, a couple ways to answer it, and we'll need to continue the conversation. I think they're sort of infrastructural questions, sort of how do you, if you want to do analyses, then necessarily need to look as far back as to the very beginning of the experiment. How do you handle that? With these distributed streaming systems, there is capability for that. With Spark Streaming, for example, you can have sort of windows of time over which you look at things, and those can extend fairly far back into the past. There's also, I guess, the sort of more analytical side or algorithmic side. Maybe there are ways of sometimes reframing analyses that we would, or sort of algorithms that we would think would need to extend all the way back in the past, but maybe don't. I think there are a lot of different ways of doing. We're finding this for a lot of streaming analyses, a lot of different ways of implementing them. I certainly think we are thinking about timescales on the order of a single experiment. So we haven't yet tackled how, like in a streaming regime, you would sort of keep track of things over months. But there's certainly plenty of companies that do that kind of thing on sort of legacy data and kind of comparing to legacy data. So basically, I think in this ecosystem, there's a lot of fun things to play with, but it'll take a lot of new ideas as well.
So I would say the same thing. Really cool, cutting edge. If everyone here wants to understand cortical circuitry, your group is among those that are building new ways to look at it. Also echo the plug for CodeNuro. I was one of those that went to your shindig in San Francisco. It was a really interesting couple days, terrific people, dazzling new technologies. So if anyone's in New York, you should certainly go. So my question, Jeremy, was what we'd like to do is understand and model the circuitry at this, the microcircuit scale. single neuron scale. I know that there's limitations inevitably in the even a great signal like GCAM6 in terms of temporal resolution.
Yeah.
So where is that today in your view? You're right where it's all getting invented. So where is temporal resolution today? What kind of a temporal relationship between an interneuron and a pyramidal cell firing can be resolved? And what's the roadmap in the next year or two?
Great question. Yeah, we're definitely sort of right at moments where things are being advanced, and there's a lot of things that are great about what we have now, but also new things that are coming. So at least the data I showed you, those examples were all using GCAM6S, which of the current, these things come in families. GCAM6 has an S and an F, slow and fast. There's also an M for medium. So S tends to have a little higher SNR, but the temporal, the kinetics, the dynamics are such that resolving the sort of timing of individual spikes is probably sort of somewhere in the range of difficult to impossible. So what you end up with is this sort of slightly temporally blurred out version of neural activity. For GCAM6F, it's definitely been demonstrated that in sort of careful sort of benchmarked experiments where you're simultaneously imaging and measuring electrophysiological signals, that you can resolve the timing of individual spikes. There are sort of algorithmic problems, how do you actually do that? And I think we need to vet and really benchmark the algorithms for doing that. But at least for GCAM6F, I think there's the potential to sort of bring it down all the way to the level of individual spikes. There will always be limitations, and certain things like adaptation and sort of change and burst rates over time can complexify that relationship. I think we'll always ideally want something faster. Developments of voltage indicators, I think, are extremely exciting. Nothing quite sort of ready for game time yet, at least for doing these kinds of in vivo experiments, but there's a lot of promising development coming from, for example, Adam Cohn at Harvard, doing some really amazing stuff, and I think that's going to be a super exciting technology. Also, I think combining tools is good. So these experiments in the mouse, Nick's been doing imaging, but he's also been doing electrophysiology. And that, of course, has a different set of constraints. You're looking now probably only over a small fraction of the population, but you are seeing individual spikes, and there's definitely right now a richness to that data that you can't get with the calcium imaging. So I think it's going to be about using the best we have, as well as kind of bringing in other tools where necessary, and sort of comparing tools and comparing what we see in these different techniques.
Thanks.
Hi, thank you for the talk. I have found when doing neuromorphic computing that the ability to visualize what the network is doing is critical to being able to say anything about it. So that's why I really like this kind of setup online and streaming. So for myself, and I assume many people here who are generating large sets of data and hope to visualize it in some insightful way, what are the sort of attributes that Spark needs to know about the data prior to be able to incorporate it into some visualization algorithm, or can it learn that on its own? Or so yeah, I think that'd be that'd help to know that if you could clarify that.
Sure. Let me just, I guess, try to ask a little bit more. Do you mean sort of like literally the format of the data, or more sort of conceptually, like what kinds of data can we?
Yeah. So binary and
like
n-dimensional matrices or something.
Sure. Yeah. So we've been doing a lot. Again, this is Spark itself, not necessarily engineered for these purposes. So it handles a lot of inputs, but didn't initially handle some of the ones that we need. So we've done work on, for example, making it easy to read in binary, sort of flat binary data, to work with kind of multidimensional array data inside Spark. That's actually been our main goal, because that turns out to be a pretty natural way to describe a lot of these imaging data sets. So at least on that front, there are things in place. There might be things that we need to add.
Sure.
But there's a lot, whether it's in Spark itself or in the stuff we're trying to build on top of it, ways of, supporting ideally a variety. So we can do different kinds of images. We can do different kinds of sort of binary representations of matrices. And dimensional arrays, we'll have to think about what that looks like on disk, but
Sure.
I'm sure, there's always a way. Nothing is impossible.
And also, so I'm not familiar with the environment you were talking about that Spark is inside, but say I'm running on AWS. Would it be possible to incorporate that with Spark as well?
Totally, yeah. So we've sort of deployed all of this stuff regularly, we and all the other people using it, either in university cluster environments or on AWS. We have a sort of single script that, partly using some Spark scripts, that basically give you an EC2 cluster running all of this stuff immediately. So that's at least one environment in which all of this works. We're looking at other cloud-based cluster environments, but right now AWS is a pretty solid one. So that's what we do a lot of stuff on. Yeah, there's always, I mean, with AWS you have to like move the data to S3. So given that sort of one problem in neuroscience is that the data will always start at the microscope. There's sort of no way around that. We've talked about parallelizing the microscope, but that sort of doesn't make a lot of sense. You can't parallelize the brain, obviously. You can paralyze the brain. You can't parallelize the brain. So that's one limitation, but transfer rates to S3 are really fast nowadays. We can take at least one of these mouse imaging experiments and put it on the cloud in about 10 minutes. So then we're sort of ready to work there.
Oh, cool. Thank you.
Sure.
So, Jeremy, beautiful work and a beautiful correlation between the destruction of neurons. But of course, those are two-dimensional picture. I wonder your thought going next to correlate it to the neuron underneath, because I'm sure there's a lot of correlation below that as well.
Yeah, great question. So that's right. In some of these examples of doing two-photon imaging, it's in either a single plane or maybe a couple staggered planes. Obviously, you really want to do something more like full kind of volumetric imaging. What I showed in the zebrafish, that is a sort of close to volumetric technique. It is still sort of optically sectioning using planes with this light sheet imaging, but it's sort of getting closer to being volumetric. People are working on ways of doing sort of what comes closer to being true volumetric imaging. And yeah, completely agree. You can also just go really slowly and sort of image one chunk for a while and then the next and then the next.
What would be your thoughts of going to volumetric approach and your direction in your institutes or your campus, rather?
Well, so that's really, I mean, there are people that are, I would say there are just sort of developments in microscopy that need to happen and that are happening. So there are people at Stanford are working on some cool new ways of doing what sort of they call sort of fully 3D imaging, this method called light field imaging. People like Michael Broxton and Logan Grosnick, who are awesome and also very involved in this CodeNuro stuff. There's also a couple of people going to be at Genelia working on some of these methods as well. I think there are a number of things, nothing's quite sort of as established maybe as two-photon imaging is, but will, yeah, hopefully give those kinds of capabilities.
Thank you very much. Thank
Sure.
you.